Hello.
It's like looking up at a two-story building. I got, oh, okay, there's what I'm looking for. Okay, kind of a busy slide. Okay, we're still going through kind of taking a look at kinases in terms of the evolution of their structure and function. And what we're finishing off here is trying to understand how it is that protein phosphorylation ever even came about as the mechanism of choice to regulate enzyme activities. Okay? And again, what's really staggering is to understand that there was really no plausible idea about this until about 5 years ago when the Farrell lab went through and did a very, very comprehensive analysis using genome sequences for a number of organisms, and going back and taking a look and saying, okay, can we find any commonality in the structures of enzymes that were originally constitutively active, okay? And so to do that, you not only have to pick proteins that you know are constitutively active, but you also need to know the crystal structures. So that kind of narrows down the scope of all the proteins you can find, because you need to know the structures to predict or to confirm that there's a particular interaction within the protein that's required for its enzymatic activity. So the first thing that they had to do was to look at, among all the proteins out there, where are the ones where there's definitive structural data that shows that that enzyme's activity is dependent on an electrostatic interaction between a positively charged amino acid, generally arginine, and then a negatively charged amino acid, either glutamate or aspartate, and that interaction is crucial for its function. So they went through and they looked at a large number of enzymes that are out there, and what they found is that the best prototypes to look at were 3 enzymes. One is topoisomerase, which is involved in DNA gyration, and more importantly, what it's involved in is in separating strands of DNA after replication has been completed. So topoisomerase, it's an evolutionarily conserved enzyme. You find it in everything going from bacteria to humans. So that was a good one to look at. And sure enough, up here, if we take a look at the crucial part of the protein structure, there we have the bridge. We've got a lysine and a glutamic acid, positive, negative. Another enzyme they went over and looked at was the metabolic enzyme enolase. Okay? Take a look at enolase again. You go back and you find that there in the structure we've got arginine and glutamate come together. And then the final one is a protein kinase called RAF. We're actually going to get to that because it's a major component in RTK signaling. RAF is also a kinase that's very, very frequently altered in a number of human tumors, particularly melanoma. Okay? So you take a look at the RAF kinase itself, and sure enough, there again is where we have a constitutively expressed form of the enzyme, and again, aspartic acid and arginine. Okay? So that establishes the premise right here that you could go back to these ancestral proteins and find that they're constitutively active because of just that single pair of amino acid interaction. But now what you have to do is to be able to say, okay, over evolutionary time, as I move from simpler organisms to more complex ones, can we see the emergence now of an enzyme that instead of being constitutively active will now be regulated by phosphorylation? Okay? So now what you would have to predict is that the original structure here would be changed to this. And that is the negatively charged amino acid, glutamate or aspartate, would be variously replaced with either serine, threonine, or tyrosine. Okay? That means that that particular form of the enzyme now would be catalytically inactive. And the only way that you could restore its activity, but now it would be an enzyme that's regulated would be now it would undergo phosphorylation. So the phosphorylation restores the negative charge at that position, allows you to reestablish that interaction, and now you can have an inducible or a regulated form of the enzyme. So sure enough, they go through, and if you take a look at topo2, RAF, or enolase, over evolutionary time, okay, start off with topo2. We look down here in all of these bacteria, okay? They've all got E's there, so they all have a conserved glutamic acid. Now we move up, and the interesting thing here is that topo2 became regulated very early on during the evolution of eukaryotic cells. Look in yeast, and you can see that even in going from bacteria to Saccharomyces cerevisiae, we lose the glutamic acid, and now it's been replaced by a threonine or serine. In fact, they're adjacent to each other, okay? If we take a look at enolase, same thing. We start off down here. It'd be constitutively active. Now we move up into eukaryotes. You look at Drosophila humans. And now it has been switched to an amino acid that could only be activated by phosphorylation. Okay? We look at RAF, and here's what's interesting with RAF. Here's where you can see the regulation or the evolution of an isoform of the enzyme that's modified by phosphorylation within human cells. And that is, we have 3 different alleles, or 3 different genes encoding the RAF kinase, A, C, and B. The B allele, as you can see, is basically here, and it's going to be constitutively active. It has aspartic acid. But the other 2 alleles that we have, the aspartic acid's been replaced with tyrosine. So it's regulated. So the interesting thing there is that now you can take a look at this evolution of phosphorylation sites and see it even within human cells. So you don't have to go all the way from unicellular organisms to multicellular to see that change taking place. It's already occurred even within human cells. Okay? So it basically shows that what this was, phosphorylation just becomes the easiest way to go from a constitutively active enzyme to one that can now be regulated very easily. And again, most importantly, it's reversible. Put the phosphate on, the protein now becomes active, dephosphorylated, and reverts back to its inactive state. It allows cells to now physiologically control when this particular enzyme is going to be active or inactive. It now means that you can more efficiently control cell physiology based on the demand for a particular enzymatic reaction to take place. It's more economical, it's more efficient. And that's pretty rare to find in biological systems. Okay, if we take a look at kinases, look at all the kinases that are out there. They have a pretty generic structure. Okay? They're kind of shaped like a little euro. And what it means is that irrespective if you're a serine, threonine, or a tyrosine kinase, you share a common structure. They all have this little cleft. And the cleft is where the substrate is going to bind. It's also where ATP is going to bind. So all kinases have to be able to bind the recognition sites on their target proteins in order to present the particular amino acid that's going to be phosphorylated, as well as ATP. Now, what this poses is a real constraint if you want to design specific inhibitors to target unique kinases. It poses a real challenge. And the reason is, kinases all share a pretty generic structure, they share a common mechanism, and the chemical playground that you have, the chemical space that you have to exploit to design a specific inhibitor to take out a particular kinase, particularly for chemotherapeutic purposes, where you're really limited is the fact that, well, they recognize serine, threonine, tyrosine, or ATP. That really puts a pretty tight box about the types of compounds you can design that are going to take out a specific kinase. So you're going to see that that poses a real problem when you're trying to target oncokinases in cancers to be able to selectively inactivate them. Okay? But if you look at the mechanism, it's a stepwise progression. And that is all kinases utilize this. They bind their substrates first. The interesting thing is that that interaction between a kinase and its substrate is not a hit-and-run interaction. And that is they're relatively long-lived complexes. That means you can take advantage of that. That if you have an antibody specific to a kinase, you can use that antibody to purify the kinase, but also isolate all of the proteins that it's phosphorylating at that time. So it's a very, very good way. You can take advantage of this to exploit it, to identify all the protein substrates for a particular kinase in a particular cell under whatever situation you want to look at. Okay? So it's very unusual to find those sorts of stable interactions between a substrate and its enzyme. So, substrate binds, ATP is already bound, so you preload the kinase with ATP, substrate binds, and then we transport the terminal phosphate to the amino acid that's going to be phosphorylated, serine, threonine, or tyrosine, and then we replace it. And then the ADP is released, gets converted back to ATP, it goes and phosphorylates somebody else, and then your substrate's released. Okay? So every kinase goes through that mechanism. So now the next level of question is, well, what distinguishes a serine threonine kinase from a tyrosine kinase? What makes them different? Okay? And again, it's the type of question that you would only be able to address knowing the three-dimensional structures of a large number of kinases. Okay? So the one that you take a look at the top, okay? This is ERK, which is the insulin receptor kinase. It's a tyrosine kinase. And what you notice about it is it has a very deep cleft. And it has to be deep to allow tyrosine, which is aromatic and much larger than serine threonine, to be able to get into it and come into contact here with the gamma phosphate of ATP. So that's a property that's shared among all tyrosine kinases, is this deep cleft. And you can see that what it does is that the cleft is so deep that serine or threonine can never go into it far enough to be phosphorylated by the ATP. So deep cleft means you've got a tyrosine kinase. Now what about a serine threonine kinase? Well, they've got a shallow cleft. Okay? So if we take a look at those, the cleft here to bind the substrate is a lot smaller than what you saw up here with the tyrosine kinase. This is a cyclin-dependent kinase. It's a serine threonine kinase that's going to be important for cell cycle progression. Okay? But if you take a look at it, it's nice and shallow. And because of that, serine or threonine, the hydroxyl group, can fit right there. And again, it's juxtaposed to where it can be phosphorylated by ATP. That cleft is so shallow, tyrosine gets pushed back. Tyrosine is so large and it's aromatic, you actually shove it back because you can't fit in it. And so you cannot phosphorylate tyrosine. Okay? So if you take a look at this, it actually illustrates what you might consider to be intelligent design. Okay? Especially if you focus up here at this really nice specificity between tyrosine and ATP and serine threonine and ATP. See? Right there. Michelangelo, this was his inspiration in the Sistine Chapel for the creation of atoms. See? Right there. Kid you not. Look. Whoops. Tell me that's not what it's showing. Okay. Now we've got one more level of specificity we have to deal with. Now you know why tyrosine kinases like tyrosine, you know why serine threonine like serine threonine. Remember, most serine threonine kinases prefer, 80% of the time, they prefer serine. So we had to wait a couple more years to see, okay, let's take it to the next level. Why do most serine threonine kinases prefer serine? And it's another one of these little subtleties. Now we're not talking about the cleft size. Now we're talking about an amino acid that happens to be very, very near the serine that's going to be phosphorylated. So now we're looking at what the amino acid is that's going to be found in the kinase that's going to facilitate that. So it turns out, remember, most of these kinases prefer serine. Serine is smaller than threonine. Okay? The difference is the vast majority of serine-threonine kinases have a phenylalanine here. Okay? The exact position doesn't matter. Don't worry about that. The key thing is that phenylalanine is positioned in such a way that it can nudge the smaller serine over so it can be phosphorylated at that terminal phosphate of ATP. Okay? If we take a look over here, the problem with threonine is the phenylalanine is large. We've got that little extra methyl group sticking off from threonine, and the phenylalanine sits on it, and it sterically inhibits the threonine from being pushed over in such a way that it can be phosphorylated. It can still be phosphorylated by those kinases, but it's a much lower affinity. Okay? So the change in that adjacent amino acid between having phenylalanine here confers this huge specificity or preference for serine. Okay? Now, what about serine-threonine kinases that prefer threonine? Okay? Well, now when you look at that relatively small number of kinases that like threonine more than serine, it turns out what you see at the same position. We still have a hydrophobic amino acid, but now instead of being the aromatic phenylalanine, it's the hydrophobic valine. Okay? And valine actually has a structure here where it can kind of come over and it just kind of helps hold the threonine in place. So again, they didn't show you the ATP, but it would be right there. And so now the hydroxyl group on threonine can be phosphorylated. Now the problem with serine is we don't have that phenylalanine there to kind of push it over and kind of hold it in place. So the problem with serine now is because it's too small. And so serine is wobbling around too much in here because we don't have that nice phenylalanine to kind of hold it in place long enough to be phosphorylated. So the ST kinases that prefer threonine take advantage of the fact that you will find a valine in their active site instead of the phenylalanine. Okay? So the key thing that you guys can see, these are all really subtle differences. Very, very subtle preferences, but it's enough to give a huge bias for the types of residues that these kinases are going to phosphorylate. Okay? All right. Kinase nomenclature is awful. It's absolutely awful. It was never systematic. It was never organized. It was never uniform in the beginning. So kinases were named by the people who were working on them in whatever context they happened to be working on. Okay? So the thing that you're faced with when you're looking at kinases, they have all these bizarre names, and there's no consistency. And the reason is the kinases were discovered for different purposes in different laboratories, and so that's how they acquired their names. So the reason it's so confusing, and the reason we don't have a common nomenclature is, for example, some kinases were designated because, well, they were studying kinases that happened to phosphorylate a particular protein. Okay? So in this case, the kinase CK1 got its name because it's a kinase that phosphorylates the milk protein casein. So we found the first one, there's casein kinase 2. They found another one, casein kinase 2, 1, 2, okay. Or this one, GSK3. Well, that happens to be the kinase that phosphorylates glycogen synthase. Okay. So again, it got its name because that's who it phosphorylates. Other kinases were discovered because of who activates them. pKa is the cyclic AMP-dependent kinase. It gets activated by cyclic AMP. That's how it got its name. PKC is a calcium-activated kinase, so that's why it got called PKC. So now you've got this whole other slew of kinases, CDKs, that we're going to be talking about. They're cyclin-dependent kinases. So they were named not by who they phosphorylate, but what's required to activate them or turn them on. Okay? We keep coming down. Now we have some that were discovered in a particular tumor or a particular virus, typically tumor viruses. So for example, ABL was discovered in the Abelson murine leukemia virus. It's also associated with a number of human types of leukemia. SART was first discovered in the Rouse sarcoma virus. So now they get a name because of the virus in which these were oncogenes that were discovered. And then finally down here, you've got receptor tyrosine kinases that were discovered based on what growth factor they bound. Insulin, epidermal growth factor. So that's how they acquired their names. So the point is, one thing as we go through this unit is you just have to be prepared that if you're looking at a particular pathway, you're going to find all these kinases that are involved in the signaling events, and they all have this very weird nomenclature. And that's just how it is. Now, one of the things to understand, these are consensus recognition sites for these different kinases. So what's something that you notice aside from the serine or the threonine or the tyrosine? What seems to be the most prevalent amino acid up there? X. You notice there's a lot of X, and X can be anything. So what does that mean? It means that protein phosphorylation sites are pretty degenerate, all right? And what that means is that in many, many cases, they'll only be phosphorylated on a particular serine or a particular threonine. But it means that a given protein can be phosphorylated by many, many different kinases. So you're never going to find a one-to-one match between a kinase and a substrate. Instead, what you're going to find, a given protein can be regulated by different kinases at different times. Okay? It affords redundancy, but it also gives the cell multiple ways of regulating the activity of proteins that are going to be phosphorylated. Okay? All right, now we're ready to get in. We've gone through the cast of players, so now you know who's going to be showing up. Now we're going to go through the first signaling pathway from start to finish and look at what happens step by step. The pathway that we're choosing is signaling that's directed by the catecholamine hormone, epinephrine, or adrenaline. It gives you a rush. And the reason is, this is the hormone that triggers what's called the fight or flight response. So anytime you get an adrenaline rush, you feel it. Your heart starts racing, it starts beating faster, you start breathing faster, and what it's doing is it's poising you physiologically to use motor activity, muscle contraction. Well, muscle contraction requires energy. The energy is going to come from glucose. Well, there's some glucose that's stored in your muscle cells, but the vast majority of your glucose is stored in your liver, and it's in the polymeric form known as glycogen. So the easiest, the fastest way that we have to increase blood glucose levels, aside from drinking an energy booster, is to break down glycogen in your liver, secrete it into the bloodstream where it immediately gets taken up into your muscle cells, and it can be used to be metabolized into ATP. So if we take a look now, we've got this physiological response triggered by this hormone, and here's our two target cells, liver and muscle. The goal here is to increase blood glucose, or to increase glucose within the muscle cell. If we take a look at the pathway that's triggered in liver cells, we get the full Monty. Everything's going to happen here. So binding of epinephrine to liver cells is ultimately going to do two things. We're going to shut down the conversion of glucose to glycogen. We don't want to do that. And while we do that, we want to activate the breakdown of glycogen to glucose. So in the liver, to most effectively do this, we're going to activate a kinase pathway, pKa, and we're going to activate a calcium-dependent pathway. Look how they're interconnected here. Okay? It's like dominoes tripping over, and we're going to activate a branch point in the pathway very quickly. The pathway that's going to require phosphorylation, what we'll call the pKa pathway, is that we're going to activate or increase cyclic AMP. Cyclic AMP activates pKa. And now you see where pKa branches off, and it's going to do two things. It's going to come over here and it's going to directly phosphorylate the enzyme that converts glucose to glycogen, glycogen synthase. Shuts it off. Okay? Done. It's also going to come over here and it's going to activate another kinase. This is a common mechanism in signaling that's referred to as a kinase cascade. One kinase turns on another one. Usually when that happens is the upstream kinase, the one that gets activated first, like pKa, has very broad specificity. Phosphorylates substrates all over the cell. pKa probably phosphorylates more proteins than any other kinase out there. One of its substrates is this kinase here, glycogen phosphorylase kinase, or GPK. Anytime you see an acronym that has a K in it, you know you're looking at a kinase. GPK is very specific. As you're going to see, it's a very complex enzyme, and it only has one substrate, glycogen phosphorylase. That's the enzyme that breaks down glycogen to glucose. So you notice pKa has a key role in both of these processes. Okay? It directly inhibits glycogen synthase. It indirectly activates glycogen phosphorylase. There's the kinase part of the pathway. But now we get calcium involved. We've got DAG and IP3 that are also going to get activated. Remember that IP3, its sole function is to increase calcium. Well, calcium comes over here. It's like looking at a football diagram. Calcium comes over here, works with DAG, and the two of them together activate another protein kinase. This is PKC, calcium activated. Here's where you see redundancy. The redundancy is that PKC also can phosphorylate glycogen synthase. It's the same amino acid that pKa phosphorylates. So glycogen synthase is getting beat up on both sides by two kinases, either of which can shut it down. And again, it shows we've got redundancy built into this pathway. Okay? Over here, though, calcium independently can also stimulate GPK. So GPK can get switched on by calcium, and it can get switched on by phosphorylation. It's additive. They're each going to give this kinase about half its activity. So they come together, and we're getting an additive activation. And combined, we're really going to make sure GP gets turned on. But if you take a look at the liver, you notice we get everybody. All the players are going to be involved. Go over to your muscle cells now. You notice it's a little different here. The reason is muscle cells can be stimulated to contract on sort of a basal level just by nerve impulses. So you're just kind of sitting here doing this, not anything in particular. That basal level muscle contraction is being driven by nerve impulses. We don't need to use a lot of energy for that. So it turns out that a nerve impulse on a muscle cell only activates the calcium part of the pathway. So you notice that what happens here is that when you only activate calcium, all we're doing here is we're partially activating GPK so that we can promote sort of a trickle-down breakdown of glucose to glycogen. You notice we're not doing anything with a nerve impulse to stop the conversion of glucose to glycogen in your muscle cells. So you're still allowing that to happen, but you're just accelerating the breakdown of glycogen to glucose from a nerve impulse. And the reason is there's nothing here that calcium does by itself to inhibit glycogen synthase. Okay? There's no kinase activated there. GPK can only phosphorylate GP. But now what happens if we superimpose epinephrine on that? Well, you still got the nerve impulse, but now we're going to bring epinephrine into the picture. And what you now see, now we get cyclic AMP. And now we come down to pKa. So now we get pKa coming over and hyperactivating it. So we get the same thing going on here by combining these two stimuli that the liver does in response to the hormone. And the reason for that is our liver's not innervated. We don't have any nerve impulses going into our liver. So we can only rely on the hormone to do both. And now over here, it's only when we get the hormonal stimulation that we're going to shut down glycogen synthesis in our muscle cells. So what this illustrates is we can take these signaling pathways, either use them all together in concert for a particular physiological purpose, and that's in your liver because that's where you get the biggest bang for the buck. That's where you're going to get the biggest increase in blood glucose. It's from the liver. In the muscle cell, we get a much more mild response. And we can get that response either through nerve impulse stimulation, and then we can build on it by adding epinephrine to the muscle as well. Okay? So you guys can see we can customize our physiological outputs based on the circumstances that you want to get the physiological output. It doesn't have to be all or nothing all of the time. Okay? All right. Let's focus on the receptor now. So the receptor is the beta-adrenergic receptor. The natural hormone here is a compound that's called a catecholamine, based on its constituents. There's the amine. This guy over here, dihydroxyphenyl, that's catechol. So generically, compounds that bind and either activate or inhibit the beta-adrenergic receptor are generically referred to as catecholamines. The natural substrate, or the natural hormone, is epinephrine, adrenaline. But we can synthesize chemical analogs. And the analogs can differ from epinephrine in two ways. The first is their affinity. We can make analogs that can actually bind with a much higher affinity than the natural hormone. They are going to have a lower KD. We'll get to that in a minute. KD is kind of a similar concept as Km. Km was involved in transport. Km was the concentration that you needed to move something half maximally. KD only pertains to binding. We're not moving anything here, we're just binding it. So KD is the concentration of hormone to bind half the receptors on a particular cell. Lower is better. Okay? So for example, here's the KD for binding epinephrine to the beta-adrenergic receptor on frog erythrocytes. I have no clue why they chose frog erythrocytes, but they did. 5 times 10 to the minus 6 molar. Okay? Here's an analog that has a much lower KD, a much higher affinity. And this is an analog called isoproterenol. Isoproterenol actually binds better. Here's one down here, Practilol, that binds with a lower affinity, has a higher KD. So you can make analogs that will still bind, but they can do it with a higher or a lower affinity. Depends on how you design the compound. And we'll look at the determinants of that binding in just a second. The other feature of an analog is it an antagonist or an agonist? Agonists bind and actually mimic what the hormone would do. They can replace the hormone. How many of you guys have asthma and you have to use an inhaler? That's what you're using. Take a look at the label on that inhaler. And what you will find is that the active ingredient, the compound that's basically causing your bronchioles to dilate and allow you to breathe better, is that it's an agonist of the beta-adrenergic or the alpha-adrenergic receptor. It's derivative, often, of isoproterenol. Okay? Anytime you see a compound that ends in OL, you know it's a catecholamine. That's just another clue there. And what those are is that they have a higher affinity, a lower KD, so you don't have to take a huge dose of it. When you inhale it, the reason you inhale it instead of taking it through the bloodstream is that way it targets your bronchial cells immediately. It binds to them, has a very, very high affinity. You don't need much of it, and you elicit the same physiological effect that epinephrine would do. It opens your bronchioles so that you can breathe better. Okay? Now, another common therapeutic application is to use what's called an antagonist. Antagonists block the effect of the hormone. They compete with it. So when an antagonist binds to a receptor, it prevents it from doing anything. And the most common examples of these, in this particular context, are called beta blockers. And what beta blockers do is that they bind to beta adrenergic receptors on cardiac muscle cells and slow down aberrant heart rhythms. Tachycardia, where the heart races out of control and beats too fast, or arrhythmia, where the individual muscle fibers of the heart beat out of sync with each other. You want to slow it down. The way you slow it down is you basically treat the individual with an antagonist of the beta adrenergic receptor. That blocks the beta adrenergic receptors and prevents epinephrine from speeding up their heartbeat. So you can use an agonist or an antagonist for a particular therapeutic purpose depending on what you want to do to the particular receptor. Now with a beta blocker, why do you think you want to use one carefully? What's the side effect or the risk that you could pose by using an antagonist as a beta blocker? What's one of the most common concerns about if you know somebody's out there driving and they're taking a beta blocker? Their reflexes aren't exactly going to be on an A game. So one of the problems with individuals that are taking beta blockers is that there's always these stern warnings on the label, avoid driving, avoid using heavy machinery. And the reason is that you're blocking the normal epinephrine response. So if they're confronted with a sudden situation where they have to respond quickly, they can't do it. The other side effect of beta blockers is that if you take them too much or at too high a concentration, it actually slows down your heart rate so much you fall asleep. So I think a lot of you guys are taking beta blockers. Okay. It's just because it's 9 in the morning. Okay. Here's what causes KD. Remember back when we talked about the GLUT transporter and the fact that GLUT1 had a very high affinity for glucose and it didn't have such a high affinity for other sugars? Same thing. The binding pockets of hormone receptors are designed to accommodate and make very specific non-covalent interactions with their ligand. The beta adrenergic receptor is designed to bind epinephrine at a very high affinity. And the reason is amino acids that form the binding pocket. So this is a side view of the beta adrenergic receptor. Now you're viewing it like you're on the outside of the cell looking in. So here's epinephrine right there. And what you find, there are specific amino acids that surround and create that binding pocket that form a series of very, very specific hydrogen bond interactions with these constituents that are on epinephrine. It's ideally designed to make these very, very specific non-covalent interactions. Okay? You can also see that we're taking advantage of the aromatic ring because it can form hydrophobic or ring stacking interactions with these hydrophobic amino acids over here. We've got a phenylalanine here at 306, we've got another one up here at 201, and then we've actually got a tryptophan down there. So the binding site has evolved to make very specific non-covalent interactions. Well, now take a look at the structure of practolol. Okay? Practolol is an antagonist, but if you look at its structure, it still has the ring, but you notice what we've done is that we've eliminated those two hydroxyl groups. And instead what we have is an amide over here. We're eliminating the possibility to form the same hydrogen bonds that we could do with epinephrine. We've also taken that hydroxyl group here and we moved it down one methylene group. So what we've done is that this can still fit. It still has the same generic shape. It's still a catecholamine. It can still bind to the receptor, but it's going to do it at a much lower affinity, so its KD goes way up. You'll still get there, but you're going to have to use a significantly higher concentration, 21 times 10 to the minus 6 molar, as opposed to 5 times 10 to the minus 6 to bind half of your receptors. So again, just like Km, the value for Kd is determined by the number and the specificity of non-covalent interactions that you can form. Okay? It's up to the organic chemist. When you're designing these analogs, you're the one who has to decide where you're going to move these substituent groups around to basically influence the binding of the compound to its particular receptor. And that's how drug discovery takes place with GPCR, simply because you know the active site structure of virtually all of them, because they all have such similar shapes. So you can design drugs that will bind to a particular GPCR with a very high degree of selectivity and specificity, just depending on how you modify the basic ligand to begin with. Okay. What this graph is showing you is the comparison between the amount of hormone I need to bind a receptor and the amount of hormone I need to get a physiological response. Okay? So if we take a look at the y-axis, we're either looking at the cellular response, that's this curve here, or we're looking at the fraction of receptors that are bound. You notice the two curves both saturate. That makes sense because you've got a fixed number of receptors. So both of these are saturable processes because you don't have an infinite number of receptors to bind to. But you notice they don't match up. If we take a look at binding, okay, if we basically ask what is the concentration of hormone that's necessary to bind half our receptors, that's Kd. So Kd is the concentration of the hormone that binds 50% of our receptors. Why do you think we use a dissociation constant instead of an association constant? The reason is when hormones bind to their receptors, it's a very fast interaction. The forward rate constant for the interaction is very quick. If a hormone binds to its receptor and it doesn't fit well, if it has a low affinity, it very, very rapidly dissociates. It has a very fast off rate. The difference between Practilol and epinephrine in their KDs is that when Practilol binds to the beta-adrenergic receptor, it immediately dissociates. High KD. Epinephrine, when it binds, remains bound. It has a very slow off rate. So it's really the dissociation rate that limits the stability of these hormone receptor complexes. So that's why we use Kd instead of Ka to refer to these complexes. Okay? So Kd is the concentration we need to bind 50%. But look at the concentration of hormone you need to get a 50% physiological response. It's shifted way, way, way over, significantly lower. So what does that mean? It means that to get a physiological response to a hormone, we bind way less than 50% of our receptors, far fewer than that. So you will very rarely ever see the concentration of a hormone in the bloodstream ever getting near its KD. You don't ever need to get that high. And the reason is you're going to see a vanishingly small amount of hormone, just a little trickle, just a whiff of it, is enough to bind a small number of receptors so that that binding is going to trigger this huge signal amplification inside the cell after it binds. And then what you're going to see very quickly is that the binding of one molecule of epinephrine to one beta-adrenergic receptor is going to cause the generation of 10,000 cyclic AMP molecules. So you go from one molecule of the hormone to 10,000 molecules of cyclic AMP from a single binding event. Your signal just went up 10,000-fold before it ever reaches the cytoplasm. That amplification takes place at the plasma membrane. Okay? So here's where we're going to piece it together then. Here's the resting state that we're going to start with. The hormone's hanging out, it's by itself. There's no hormone there yet, so the receptor's off. Our trimeric G protein complex is also off. You notice integral membrane protein, lipid-anchored protein. So these guys are covalent lipid anchors that only insert into the cytosolic leaflet of the plasma membrane. So they're right there inside the cell, ready to be activated. But it's off because it's got GDP bound. Here's who we need to switch on. Another integral membrane protein, adenylate cyclase. This is the guy that's going to convert ATP to cyclic AMP. But the hormone binding to the receptor can't do that. We have to go through the G protein. The G protein's off. So to switch it on, we need a GEF. We need a GEF to replace or exchange GDP with GTP. Well, here's the good news. Guess who our GEF is going to be? The hormone receptor. All we've got to do is bind the hormone. There's our GEF. Okay? So here's how it happens. To get the process started, binding of the hormone out here. Now remember, the hormone never goes into the cell. Look at the conformational change in the receptor by binding that one little molecule of epinephrine out here. It basically causes the cytosolic regions of the receptor to go like this. It basically opens up. And when it does, it binds to the G protein. So it captures it. It literally captures the alpha subunit and it pries it open. And it pries open the alpha subunit to let the GDP diffuse away and now can be replaced with a molecule of GTP. That's it. Now you're off at the races. So what happens next? Here's where we're going to get our first amplification. So just to finish up for today, here's what's going to happen. These come over here and they interact. The hormone receptor complex acts as a GEF. We exchange GDP for GTP. Now, the G protein complex dissociates. Alpha goes its own way. Beta, gamma are still hanging around. Don't worry about them. Alpha GTP now interacts with adenylate cyclase. And as you're going to see on Friday, that's what we need to get adenylate cyclase active. The key thing here is, notice, guys, we're going from one protein-protein complex to a new one. We're taking a protein-protein complex between GS alpha and G beta gamma. That's the resting state. The active state is G alpha GTP. Let's go of these guys, and now it binds to adenylate cyclase. A change in protein-protein interaction. Here's the amplification we get. One hormone receptor complex activates 100 of these. Remember, they can diffuse laterally in the plasma membrane. So as soon as one hormone binds one receptor, it pings over, hits this G alpha complex, exchanges GDP for GTP, and then it lets go. Well, that means our receptor can slide over and ping into another G protein. For the duration that the hormone is bound to the receptor, one receptor activates 100 G proteins. 100-fold amplification right there. First step, 100-fold increase in your signal. That one G alpha, now it has to hook up with one adenylate cyclase. It's 1-to-1. They have to stay together. But we activated 100 G alphas. So that means we can activate 100 adenylate cyclases. Cool with that. Here's the final number. Each activated adenylate cyclase, before this guy hydrolyzes GDP to GDP and shuts off, will synthesize 100 cyclic AMPs. There you go. 10,000 cyclic AMPs from one hormone binding to one receptor. 10,000-fold right there. So where we're going to pick it up on Friday, though, is using a really cool technique using fluorescently tagged proteins to follow the protein-protein interactions. Okay? So that's where we'll start it on Friday.